Today I'll be sharing some results of the characterization of defects in midline bone and cartilage patterning associated with HYL2 deficient mice. So HYL2, or hyaluronidase 2, takes these high molecular weight HA fragments and degrades them into intermediate sized pieces, which can then be endocytosed and further degraded at the lysosome. During bone development, mesenchymal cells will migrate into a cell-free, HA-rich environment where they can condense and proliferate. These cells will then differentiate as HA is removed. Bones can be made in two different ways: through intramembranous ossification, where these mesenchymal cells will differentiate directly into these pre-osteoblast cells, or through endochondral ossification, where they become chondroblasts first. RUNX2 and Osterix are two important transcription factors that allow these cells to differentiate into osteogenic lines. Now, humans with HYL2 deficiency have a number of bone deformities, which include cleft palate, as well as pectus excavatum and conductive hearing loss. So our main objectives for this study were to characterize the skeletal phenotype of HYL2 deficient mice. We also wanted to determine how HYL2 deficiency, or increased HA through development, impacted cellular condensation, proliferation, and differentiation of bone cells. So to do this, we generated HYL2 deficient mice by intercrossing HYL2 heterozygous mice to generate embryos of 17.5, 18.5, and 19.5, as well as neonatal pups. Animals were then whole mount stained with Alcium blue and Alzarian red. Micro-CT imaging was done at 9 micrometer resolution, and various histological staining was conducted using Goldner's trichrome Alcium blue staining, immunohistochemistry with anti-Osterix antibody, and any measurements of histology were taken with ImageJ software. Micro-CT and whole mount staining revealed abnormalities such as delay in ossification, central widening, and other patterning defects. When examining bones that undergo intramembranous ossification, the commonly affected bones were those associated with the palate. These areas were often widened with decreased ossification. They also were associated with decreased staining of Osterix. When examining bone formed from endochondral ossification, commonly affected bones were those in the sternum. The chondroblasts here are in an earlier state of proliferation and have a less defined periosteal layer. These studies suggest that HYL2 plays an important role in the removal of HA to promote condensation of pre-osteoblasts or pre-chondrocytes during bone development.